Okay. Welcome, my dear friends. Within 3 days, we're going to sum up our CPGET free online coaching. So, eminent and huge experienced resource persons has delivered their lectures. In this regard, today's our most experienced and eminent personality, Dr.B.Rukmini Devi Madam Garu, Assistant Professor, Head Department of Botany, GDC Narsapur, Medak District. We would like to invite respected Madam Garu. Please welcome. And
thank you, sir. Very much, sir.
Share your screen. I have given to you and start your session, madam. Please welcome.
Okay, sir. Thank you, sir. Good afternoon, sir. Am I audible, sir?
Yes, madam. Yes, please, madam, please proceed.
Okay, sir. Thank you, sir. Thank you for your welcoming words. Good afternoon, everyone. Today I am going to deliver a lecture on life cycle of Mucor. I hope all of you know what is Mucor, to which subdivision it belongs to. Okay. Actually, Mucor is a fungus. First, we will start its life cycle with systematic position of this Mucor. Mucor belongs to division Eumycota. It belongs to the subdivision Zygomycotin, and class it belongs to Zygomystes, and order it belongs to Mucorales. In the family it belongs to Mucoraceae. The genus Mucor, which includes almost 50 species. How many species are included in the genus Mucor? It includes almost 50 species. This Mucor genus, commonly called as pin mold, or also called as black mold. Most of the species are saprophytic in nature. What kind of nature these will show? These will show saprophytic nature. That means these are growing on dead and decaying organic matter. These are not only growing on dead and decaying organic matter. These are also grows on variety of substrates. For example, food materials they will grow. They'll grow on the substrate of soil. They also grows on the substratums which are in contact with the soils. Mostly this Mucor fungus will prefer damp conditions for its growth. Mostly these Mucor members are growing as saprophytes. They are growing on variety of habitats. Okay. What kind of habitats they will grow means they'll grow on the damp soils. They'll grow on the food materials. They'll grow also on the substratums which are having close contact with the soils. They are also growing on the some organic materials, for example, jams, jellies, and overripe fruits, syrups, and bread. Mostly these members are growing on the organic materials. So here you can see the Mucor, which is growing on the bread. Okay. Common name for Mucor is black mold or pin mold. These are the common names for this Mucor. This Mucor is not only growing as a saprophyte. Some of the species which are growing on dung. For example, Mucor mucidos species and Mucor himalis species. These two species are simply described as coprophilous fungus. Why these are called as coprophilous fungus? Because they love dung for their growth. On the surface of the herbivorous animals' dung, these species, especially Mucor mucido and Mucor himalis species, will grow. That is why these two species are called as coprophilous species. Copro means dung, philous means loving nature. So they will grow on the surface of the dung. On what type of animals dung these will grow? These will grow on the herbivorous animals dung. That is why these two species are called as coprophilous fungi. Some of the species which are growing as laboratory contaminants. Examples for this kind of laboratory contaminants are Mucor racemoses species, Mucor racemoses and Mucor mucido. These two species, one is Mucor racemoses and another one is Mucor mucido species. These two species are growing as contaminants in the laboratories. Some of the species are known to cause one disease that is called as mucormycosis disease. Okay, what type of species are causing this mucormycosis disease? Mucor mucido species will cause this disease. Actually, this disease is caused by Mucor mucido species. This disease is identified in human beings as well as domestic animals. This is actually a serious disease and fatal disease in some patients which are using some immunosuppressive drugs. In such kind of patients, this mucormycosis disease is fatal. This is about different habitats of mucor and what kind of harmful effects it is showing, we have discussed. But some of the species of mucor are industrially important. Why these are industrially important means, here these mucor species are used in the production of fermented products, for example alcohols, etc. What type of species are used in this industries means, one is Mucor indicus species and another one is Mucor circinelloides species. These two species of mucor are used in the industrial production of some fermented products, for example alcohols, etc. Mucor species can be grown very easily in the laboratory by placing bread slices. Okay, suppose these bread slices are moist in condition, means suppose they are having moist nature, then in the moist chambers these mucor species will grow. In the laboratory conditions we can grow this mucor species very easily just by keeping some bread slices in the moist chambers. At room temperatures only this mucor species will grow in the form of white cottony tufts. Tufts means just bunches in such a form, these will grow in the laboratories also. At room temperature only these will form. These are actually produced in white cottony tufts. In group these are appears white in color, but individual mycelium is appears hyaline. Hyaline means colorless hyphae. Hyaline means colorless in condition. This is about habitat and the number of species which are identified in mucor. Now we will discuss about the vegetative morphology of the mucor fungus. Actually, this mucor belongs to Zygomycotina subdivision. Here, the plant body is in the form of thallus. So, thallus means here plant body is in the form of mycelium. How this mycelium is present? Actually, mycelium is made up of several filaments. This mycelium is white in color and it forms a cottony substrate. Okay, on the surface of the substratum, this mucor is growing as a white cottony surface. Okay, here this mucor thallus, which is present in the form of mycelium, which is a branched mycelium. That means mycelium is showing branches. Here mucor mycelium is a cenocytic in condition. Cenocytic condition means here in the cytoplasm many nuclei are present. Here this mycelium is not divided into chambers by septa. There is no septa formation in this mycelium. So many nuclei are present in the cytoplasm. So that kind of aseptate condition with many nuclei is called as a cenocytic condition. So mucor thallus is showing Cenocytic condition. So vegetative structure of this fungus, if you observe, here you can see the structure of this Mucor fungus vegetative plant body. Okay, from this vegetative plant body, some reproductive structure is also produced here. Sporangiophores you can see in this image. So how this vegetative structure will produce as reproductive structures we will discuss while discussing about reproduction and the types of reproduction. First we will discuss about vegetative morphology of the fungus Mucor. In the form of groups, okay, whenever it is present in the form of a colony like kind of conditions, it is white in color. Okay, actually it is a cottony in appearance, white in color. But whenever if you observe individual hyphae or hyaline means colorless in condition. Sometimes due to the formation of these spores, this Mucor mycelium may appear grey in color or brown in color or black in color also. Why these colors are formed means due to the formation of spores, this Mucor fungus is appearing in different colors, white, black, brown or grey in color. Actually, this mycelium is aseptate cenocytic in condition until formation of reproductive structures. Whenever reproductive structures are produced, then septa formation will take place. So unless and until the formation of this reproductive organs, the mycelium is aseptate mycelium which is showing branches. Here this mycelium is a cenocytic in condition, we have discussed already. Cenocytic means many nuclei are present in the cytoplasm, but there is no wall formation or septa formation. Here if you observe this mycelium, here this mycelium will show two types of growth. One is prostrate growth and another one is erect growth. Prostrate mycelium which are buried deeply in the substratum, and this prostrate mycelium helps in two main functions. One is absorption of nutrients from the substratum and another one is giving mechanical support, just like anchoring. Okay, how the roots are giving support to the plants in such a manner, here this prostrate mycelium will give support just like an anchor. So mainly this prostrate mycelium performs two functions. One is anchorage function and another one is absorption function. What it will absorb? It will absorb nutrients. Mineral nutrients are absorbed by this prostrate mycelium of Mucor from the substratum. What is another type of mycelium which is also developed during its growth means that is erect mycelium. When this erect mycelium will develop means especially during the reproduction process, this mycelium will grow aerially in erect manner. So from this erect hyphae, this reproductive structures sporangiophores and sporangia will develop. So two types of mycelial growth we can see in this fungus Mucor. One is prostrate mycelium and another one is erect mycelium or aerial mycelium. Okay, here in vegetative stage, this hyphae consists some structures, those are called as a stolon. Okay, here cell wall of this Mucor, if you observe, it is made up of chitin and chitosan material. Here cellulose is totally absent. Okay, this mycelium cell wall is made up of two main substances. Chemically, this cell wall is made up of chitin and chitosan material. Cellulose is not present in this Mucor. If you observe the cytoplasm of this Mucor cell, what kind of cell organelles are present in its cytoplasm means mitochondria are present, endoplasmic reticulum is present, next ribosomes and vacuoles also present. You can see all these cell organelles in the cell of mucor. This is about morphology and structure of mucor mycelial cells. Here you can see this is the cell structure in mucor. Here in the cytoplasm many nuclei are present. This cell is surrounded by plasma membrane. This plasma membrane is surrounded by cell wall. This cell wall is made up of chitin and chitosan materials. In the cytoplasm you can see nuclei. Next you can see cell organelles like endoplasmic reticulum, mitochondria, ribosomes, vacuoles, etc. So this is about structure of cell in mucor mycelium. Here one important point I want to mention here. Actually this mucor, it is mycelial form. Plant body is mycelial in condition, but sometimes this fungus mucor is produces yeast-like structures. This condition is called as torula state. Okay, sometimes this fungus is appears in yeast-like in condition. This condition is called as torula condition. When you can see this kind of torula stage means whenever this mucor is growing on the liquid nutrient mediums under laboratory conditions. Suppose if you are growing this mucor in laboratory on liquid nutrient medium, then conditions are semi-anaerobic conditions. In what kind of environmental conditions you are growing this fungus? Semi-anaerobic conditions. If you are growing this mucor in semi-anaerobic conditions and you are growing this mucor on nutrient medium. What type of nutrient medium? Liquid nutrient medium. Okay, then what will happen? This mucor appears in yeast-like structure. This stage is called as torula stage. So this mucor appears in two states. One is mycelial state, and mycelial means filamentous state, and another one is yeast-like state. All of you know how the yeast cells are present. Yeast is a unicellular structure, unicellular fungus. Okay, sometimes by budding these yeast cells are produced continuously and all are maybe arranged in the form of a filament, means that is called pseudomycelium. You will see that pseudomycelium formation in yeast, but actually yeast is a unicellular fungus. So how the yeast cells are present in such a manner, here mucor is growing in laboratories. That state is called as a torula state or torula condition. So whenever is not sufficient, whenever oxygen is not sufficient, in such situation this mucor develops into yeast-like structures. So mucor is having one special ability that is called as switching between yeast-like state and filamentous state. So because of this ability, this mucor is also called, mucor is described as a dimorphic fungi. Why it is called as dimorphic fungi? Because it is having ability to switch between two different states. One state is filamentous state, that is common state. Mostly it is appears in filamentous state. But another state is, it is appears in the form of yeast-like state. So it is having ability to switch between yeast-like state and this filamentous state. That ability is called as dimorphism. That ability is called as dimorphism. Okay, so this is the important point what exhibited by mucor I want to mention here. Next we will discuss about the reserve food materials, how the food materials are stored in the mucor cell. Food materials are mostly stored in this mucor cell in the form of glycogen as well as oil globules. How the food materials are stored in mucor cell? The food materials are stored in two forms. One is glycogen granules and another one is oil globules. In this form, food materials are stored in mucor cell. This is about vegetative morphology of the fungus Mucor. Next, we will discuss about reproduction. How the reproduction will take place in Mucor. Actually, Mucor reproduced by three methods. One is vegetative reproduction, and another one is asexual reproduction, and third one is sexual reproduction. So, Mucor reproduced by three methods. One is vegetative, and another one is asexual, and the third one is sexual reproduction. How Mucor reproduced vegetatively means this vegetative reproduction takes place by three methods. One is fragmentation method. Second one is chlamydospores formation. Third method is formation of oidea. These are also called as arthrospores. By these three ways, this Mucor will reproduce vegetatively. We will discuss all these three methods in a detailed manner now. What is the first method of vegetative reproduction? One is called as fragmentation. Fragmentation means here the mycelium or the filament breaks into small pieces. Whenever conditions are favorable, each piece germinates into new mycelium. That kind of vegetative reproduction is called as fragmentation type of vegetative reproduction. Okay, first one is fragmentation. Fragmentation means it is nothing but breaking of this filament or mycelium into small fragments or pieces. Each piece further develops into new mycelium. Whenever conditions are favorable, each piece or each fragment germinates and giving rise to new mycelium or new hyphae. That is called fragmentation method of vegetative reproduction. Next, second method of vegetative reproduction is chlamydospores formation. When these chlamydospores are produced, whenever conditions are unfavorable, that means during adverse conditions, this chlamydospores formation will take place. How these chlamydospores are produced means actually the mycelium consists of many cells. So these cells are surrounded by thick walls. These thick walled structures are called as chlamydospores. By developing thick walls around these cells, these are escaping from the adverse conditions like desiccation, etc. So these cells are containing food materials in huge quantities. So the food materials are gets accumulated in cells and they will surrounded by thick walls. So by these thick walls, they can escape from the adverse conditions like desiccation. Then these are in resting state. So like this, they are escaping from the adverse environmental conditions by developing these thick walls around the cells. By using the food materials, they will escape from the unfavorable conditions. When conditions are favorable, whenever conditions are favorable for the growth, then this wall will be ruptured and the chlamydospore will germinate into germ tube and that produces mycelium. Like this fungal mycelium develops thick walled cells. Those are called chlamydospores. By producing this chlamydospores, this fungus is escaped from the adverse environmental conditions. Once conditions are favorable for the growth, then these chlamydospores started germination. Then by germination, these will produce new mycelium. Like this, Mucor reproduced by chlamydospores in unfavorable conditions. Next, what is the third method of vegetative reproduction? That is called as oidea formation. In some of the fungal members, cells of the mycelium divides and forms round-shaped structures. Okay, how these are formed? The cells of the mycelium divides and they will develop into round-shaped cells. These cells are developed in a series. Okay, that kind of structures are called as oidea. These are also called as arthrospores. These cells are develops into round shaped structures. These are arranged in a series, continuous series form. These are arranged. These are behaves like spores. That kind of structures which are developed in a series, those are called as oidea. These are also germinates whenever conditions are favorable. Then they will give rise to new mycelium. Like this, mucor reproduced vegetatively by oidea or arthrospores. This is about vegetative reproduction in mucor. Next, we will discuss about asexual reproduction in mucor. How mucor reproduced asexually? Actually, this mucor is reproduced asexually by the formation of one specialized structure that is called as a sporangia. Okay, when this mucor is reproduced asexually whenever conditions are favorable, then mucor reproduced asexually. So what type of structure is produced during this asexual reproduction? Sporangium will develop during this asexual reproduction. How this sporangium will develop? Actually, if you observe this diagram, you can understand how this asexual reproduction will take place. From the mycelium, some mycelial parts are growing aerially. Actually, here you can see aerially growing mycelium and prostately growing mycelium in this diagram. So, this prostately growing horizontal mycelium helps in fixation onto the substratum as well as absorption of nutrients from the substrate. But some of the mycelial branches are growing aerially. Growing aerially means growing above the substratum. Okay. So, these are growing in erect manner, straightly, in vertical manner these are growing. That kind of vertical branches which are growing aerially in erect manner, that kind of vertical branches are called as sporangiophores. Those are called as sporangiophores. So, from this mycelium, this kind of sporangiophores will develop. Later, the contents, whatever it is present in this sporangiophores, contents means here cytoplasm, nuclei, etc. These contents which are gets accumulated at the tip region of the sporangiophore. What will happen? At the tip region of the sporangiophore, the cell contents are gets accumulated. So what will happen? The tip region will become swollen in nature and becomes a round in shape. Okay, that swollen tip region which becomes round in shape, that is called as a sporangium. So, from this elongated filamentous tube-like sporangiophores, tips are swollen due to the accumulation of cell contents. So, as a result of this accumulation of cell contents in the tip region of sporangiophores, these tips are enlarged in size and round in shape. These structures are called sporangia. These structures, ball-like structures, are called as sporangia. So, later, the contents, whatever it is present in this sporangium, are differentiated into two regions. One is peripheral region and another one is central region. The contents, cell contents are distributed unequally to the peripheral region as well as central region of this sporangium. So what will happen? In the peripheral region, excess amounts of this nuclei, cytoplasm will be accumulated. But in the central region, only less amount of cytoplasm, less amount of nuclei will be accumulated. So as a result, here one special structure which is formed here between this peripheral region and central region and separating these two regions by this special structure. What is that special structure produced inside this sporangium which is separating these two regions? That is called as a columella. One special structure which is developed here, that is called as a columella. That columella is developed here. That columella is separating this peripheral region from the central region. Actually, this columella is a semicircular shaped structure or dome shaped structure. You can see here in this diagram, you can see columella. See right side image, you can see columella. So one dome shaped structure or semicircular shaped structure will develop inside this sporangium. That columella, which is separating this peripheral region from the central region. Once this columella is formed, later this columella is separating two portions, peripheral portion and central portion. Later what will happen? Here on maturity, what will happen? Here the sporangial wall will be ruptured. What will happen? The sporangium is surrounded by wall layer, no? So that wall layer is dissolved. So due to the dissolution of this sporangial wall layer, what will happen? Inside this peripheral region, this cell contents are divided into small round shaped structures. Those round shaped small structures which consist cytoplasm and nucleus, those are called spores. So before wall rupture, what will happen? The cell contents in this sporangium differentiated into spore-like structures. Before this cell wall dissolution, what will happen? The cell contents which are present in this peripheral region, those are differentiated into round shaped structures. Those are called as spores. Each spore consists of some amount of cytoplasm and one nucleus. So like this sporangium cell contents are differentiated into many spores. So these spores are formed inside this sporangium and they will increase in size. So whenever the spores which are produced inside this sporangium, whenever these spores are reaches maturity, then what will happen? The sporangial wall will rupture. Then these spores are released from this sporangium. What will happen? Here first sporangiophore will develop vertically from the mycelium. From this sporangiophore, at the tip region, cell contents are accumulated. So as a result, the tip will become round in shape and swollen in condition. This swollen round shaped structure is called sporangium. In this, cell contents are differentiated into two parts. One is peripheral part and another one is central part. In the peripheral region, many nuclei are accumulated and dense amount of cytoplasm also accumulated. More amount of cytoplasm is accumulated and many nuclei are also accumulated in the peripheral region. That means outermost region. But in the center region, less amount of cytoplasm is present and less number of nuclei are accumulated. So this center part is differentiated into columella region. Center part is differentiated into columella region, which is a dome shaped structure or semicircular structure. Later, what will happen? Inside this sporangium, cell contents are there, no? Those are differentiated into, differentiated means the cell contents are divided into small pieces. Cell contents are divided into small pieces. That means one nucleus, some amount of cytoplasm, and that is differentiated into one spore. Like that, many spores formation will take place in the sporangium. Like this, many spores are produced inside this sporangium. These spores are developed well. Whenever these spores are reaches maturity, what will happen? The sporangial wall will breaks. So once the sporangial wall is breaks, then the spores which are developed inside the sporangium are released into the environment. So like that, many spores which are developed inside this sporangium are released into the environment. These are actually non-motile spores. What type of spores these are? These are non-motile spores. That is why these are also called as aplanospores. The spores which are produced during asexual reproduction are called as aplanospores. Aplanospores means these are non-motile spores. They do not possess any flagella, so they cannot move from one place to another place. So how these are distributed or how these are spread to different regions with the help of wind? These spores are liberated from this sporangia and they will spread to various regions. So these are non-motile spores. That is why these are named as aplanospores. Like this, many aplanospores are produced from this sporangium and they are released into the environment. Later, these spores fall onto the suitable substratum. Then they will germinate and giving rise to germ tube. Finally, they will produce mycelium. Like this, Mucor reproduced asexually. Here, the spores which are produced by this asexual reproduction are haploid spores. So, they will give rise to haploid mycelium. So, here, asexual reproduction results in the formation of a large number of haploid spores which are develops into haploid mycelium. Like this, Mucor reproduced asexually by these spores. Here, on maturity, these spores turns to black color. That is why these are also called as black molds. These Mucor species are also called as black molds. This is about asexual reproduction in Mucor. Here you can see the liberation of spores. Here you can see the clear structure of these spores, how these are appears at the tip of the sporangiophore. Okay, you can see here sporangiophores and at their tips sporangia are there. Finally, on maturation, these spores are separated from this sporangium by the rupture of sporangial wall. Whenever conditions are favorable, these spores fall on, whenever these are falls on the substratum, then they'll start germination and they will produce first one germ tube. Later, these are develops into mycelium. Like this, Mucor reproduced asexually by the formation of spores. Okay, this is about asexual reproduction. Now we will discuss about sexual reproduction in Mucor reproduced sexually. What type of sexual reproduction it will show, we will discuss here. Actually, Mucor reproduces sexually in unfavorable conditions. Whenever conditions are not favorable for their growth, then it reproduced asexually. By what method it reproduced? Sorry, whenever conditions are unfavorable, it reproduced sexually. So, by what type of method it reproduces sexually means gametangial copulation method. It reproduces sexually. What is this gametangial copulation? We will discuss now. Mucor reproduced sexually by gametangial copulation method. In unfavorable conditions, it reproduced by this method. Here, before discussing about this gametangial copulation, we have to discuss one important phenomenon here that is called as heterothallism. What is this heterothallism? Who discovered this phenomenon? This heterothallism phenomenon was first discovered by A.F. Blacksley. He is an American geneticist. Who is he? A.F. Blacksley. He is an American geneticist. He discovered this phenomenon of heterothallism. What is this heterothallism means? Actually, mating takes place between two compatible species. Suppose these two species are two male and female which are involving in this reproduction process, which are arising from the same species, which are compatible to each other and which are able to perform Sexual reproduction means those are called as homothallic species. Here we have to discuss about homothallic species and we have to discuss about heterothallic species. What is homothallic? What is this homothallism and what is this heterothallism? We have to discuss. Who discovered this heterothallism? A.F. Blacksey. He is an American geneticist. He discovered this heterothallism phenomenon in Mucor mucido species. In which year he discovered this phenomenon? 1904 he discovered this phenomenon. Actually, in this Mucor mucido species, here two species are involving in this sexual reproduction process, but the two species which are involving in this sexual reproduction process, these are not different morphologically. That means these are morphologically similar species. These are morphologically similar species. And these morphologically similar species are producing two different structures. These structures are called as gametangia. Actually, the structures which are involving in this sexual reproduction process of Mucor, those structures are called as gametangia. That is why here the method of sexual reproduction is called as gametangial copulation. Here the structures which are involving actually in the sexual reproduction process are called gametangia. Here in this Mucor mucido species, the two gametangia which are involving in this sexual reproduction process, those two gametangia are morphologically similar. That means there is no difference present between these two Mucor mucido varieties. These two are compatible to each other. That means they can fertilize with each other. These two are morphologically similar but physiologically different. That type of condition is called heterothallic condition. Simply heterothallism you can define like this. How to define heterothallism in simple words? The presence of morphologically similar species, and these similar species morphologically are different physiologically. That kind of condition is called as heterothallism. You can observe this heterothallism in almost all groups of fungi. This heterothallism you can observe in all groups of fungi. So here the mating occurs between two morphologically similar species, but these two morphologically similar species are differs in their physiological processes. So here you cannot differentiate this morphologically similar species into male and female. So whenever sexual reproduction will take place, whenever one male structure and another female structure involving in this process, then you can describe that process as a sexual reproduction. But here the fusion takes place between two structures, but these two structures you cannot differentiate them morphologically, but physiologically can differentiate them. That phenomenon is called heterothallism. So here these two strains you cannot differentiate them as male and female. So you can differentiate them as, or you can describe them as plus strain and minus strain. So here the two strains which are developed from the single mycelium, which are similar morphologically but physiologically they are different. Between such kind of two fungal mycelium, some specialized structures will develop. Those specialized structures here you can see. See here two strains you are observing the first image. From this mycelium, some specialized swollen structures are developing here. Those are called as Progamitasia. Those are called as Progamitasia. That means here the mycelium consists of cell contents. Cell content means cytoplasm, nuclei and other structures. These contents are accumulated to the tip region. So the tip regions are slightly curved and rounded off. They are swollen in condition. This is the initial stage of gametangial development. That is why that is called Progamitosia. Initial stage of Gamitangia is called Progamitosia. So in the beginning, from this plus strain of mycelium and minus strain of mycelium, two swollen structures will be developed from this fungal mycelium. Those are called Progamitosia. Later, this Progamitosia enlarges in size and becomes round in shape. Later, these swollen portions are separated from the basal portion by the formation of one wall. In the beginning, I told you, unless and until the formation of sexual reproductive structures, this Mucor mycelium is accepted. So here, once this Progamitosia formation taken place means just below this Progamitosia, one septa will form. That septa separates this Progamitosia from the basal portion of the mycelium. So this enlarged portion of the Progamitosia increases in size by the accumulation of cell contents. So this round shaped enlarged structures are called as Gamitangia, and the basal regions of this Gamitangia are called as suspensors. Basal region of this Gamitangia are called as suspensors. So like this, two different strains are produced, two Gamitangia. These two Gamitangia are growing towards each other. For this, some hormones are required. This was reported by another American biochemist. Those hormones are inducing the development of this Gamitangia. That scientist name is Burgess. He identified this hormone's role in the formation of Gamitangia and their fusion. He is American biochemist. He is American biochemist. His name is Burgess. In 1925, he reported that some of the hormones are playing important role in this Gamitangial formation and their fusion. Those hormones are secreted by this two fungal mycelium, and they are inducing them to involve in this fusion process. That hormone name is trisporic acid. Which is important role here? Important role here. This trisporic acid stimulates the Mucor mycelium to participate in this Gamitangial population process. So, like this, two strains produces two Gamitangia. These are enlarged in size. So now these are called as Gamitangia. In the initial state, these are called as Progamitosia. So once these are increases in size and reaches maturity, these are called as Gamitangia. These two Gamitangia are coming close to each other and growing. So once these two Gamitangia touches each other, two strains Gamitangia coming close to each other, growing towards each other. So this left side one is going towards right side one is going towards left side. Like that, two Gamitangia which are produced from two different strains which are growing towards each other. So at some state, these two Gamitangia touches each other. At the point of contact, what will happen? This Gamitangial walls will be dissolved. So what will happen? The contents, whatever it is present in these two Gamitangia, are mixed with each other. The contents, that means the cytoplasm, nuclei, and cell organelles which are present in these two Gamitangia are mixed with each other. So as a result, one structure will develop. That structure is called zygospore. How this zygospore is produced? Third diagram, you can see this zygospore. The two Gamitangia are contact with each other. At the point of contact, their walls will be ruptured or dissolved. Due to this dissolution, their contents will be mixed with each other. So as a result, one structure will develop that is called zygospore, which is a round shaped structure or spear shaped structure. This zygospore is surrounded by two wall layers. Outer layer is spiny in nature, inner layer is thin in nature. This outer layer is called exosporium, inner layer is called endosporium. So like this, with two wall layers, this zygospore will be protected. So like this zygospore formation will take place by this gametangial copulation method of sexual reproduction. So once the zygospore is formed, then what will happen? It will undergo resting phase for few months. It will undergo for resting phase for few months. So once it completes its resting period, then this zygospore starts germinate. How it will germinate? This wall which surrounds this zygospore will rupture. So this zygospore will be growing vertically. Here you can see, in this image here you can see, this zygospore is growing vertically and develops into a filamentous structure. That is called as a zygosporangiosphore. Okay, this elongated vertical structure which is developed from this zygospore during germination, that is called as a zygosporangiophore. This elongated structure develops and produces one structure that is called germinating zygosporangium. Like this, zygosporangium will be produced from this zygospore. Later, the contents, whatever it is present in this zygosporangium, those are diploid in condition because two gametangia contents are mixed, no? So here diploid condition arises in zygosporangium. So the diploid nuclei, whatever it is present in this zygosporangium, undergoes meiotic division. So haploid spores will be formed. Whenever this zygosporangium wall will be ruptured, this haploid spores will be released from this zygosporangium. With the help of wind, they will be reached various substratum and they will start germinate in favorable conditions. So they will produce mycelium again. So like this, they will start their new life cycle. So like this, Mucor reproduced sexually by gametangial copulation method. So this is about sexual reproduction. If you observe life cycle of Mucor, haploid phase is dominant. Diploid state only you can see after this gametangial copulation. So it is a short-lived phase. Diploid phase is a short-lived phase, but remaining time it is present in haploid state. That is why life cycle of Mucor is described as haplontic life cycle. So with this, I want to conclude this session. So in today's class, we have discussed about life cycle of Mucor. In this, we have covered habitat of Mucor, vegetative morphology of Mucor, and three different types of reproduction which occurs in Mucor: vegetative, asexual, and sexual reproduction. We have discussed. With this, I want to conclude. Thank you for listening. Thank you, sir, for giving this opportunity.
Thank you so much, ma'am.
Thank you, ma'am.
Sir, one doubt.
Yes, any doubt?
यस सर सर आप जो ग्रुप में भेजे नहीं वो क्वेश्चंस मतलब क्विज के टाइप में जो क्वेश्चंस भेजे हम लोग वो पढ़ लिए तो उसमें से टच होते सारे एंट्रेंस में
Yes, definitely. We'll send another 3 mock tests will send in our WhatsApp group. Are you sent your hall ticket, Amma?
या ओके सर OU
Okay.
31
Okay, no. You sent me your hall ticket hard copy. Downloaded copy, please send me in WhatsApp group. PG entrance.
SS भेजना सर
PG entrance. Am I audible? Arshia ji,
यस सर
you sent your PG entrance hall ticket.
ओके सर
Right. Madam, really wonderful presentation.
Thank you, sir.
You have mentioned all the important points, especially zygomycotina fungal member, that is a mucor, mucido importance, and mucor mycosis, one of the most pandemic situation we are very painful situations we are observed. Some of the younger people, they have lost their lives just because
Also
of this
affected by
mucor mycosis, that is a black fungus.
sir.
And
Yes,
also,
sir.
you have clearly explained about the heterothallism by A.F. Blacksley. And also, you have clearly explained about the general characteristic features of the Zygomycotina, mucor, classification of the mucor, vegetative characteristic features, morphological features, vegetative reproduction, and asexual reproduction, even though sexual reproduction, life cycle. Clearly explain each and area, each and every corner of the point you have mentioned this class, madam. Really wonderful presentation. I hope your classes enjoy lot. Our young friends definitely will benefit from your class, madam. Really, thank you very much, madam.
Thank you,
We'll
sir.
meet,
Thank you so much for giving your.
yes,
you.
madam. Yes, you have covered all the points. Thank you.
Thank you.
Right, madam. Right.